Well, ladies and gentlemen, may I first of all acknowledge the traditional custodians of the land upon which we are gathered today, the Kombumerri people. May I also acknowledge the Chancellor, Lenine Ford AO, Mr. Terry Robertson, Doctor of the University, Chair of the Gold Coast Advisory Council, and Deputy Chair of the Institute's Board of Advice, a lot of titles. Ms. Suzanne Gallagher, representing the Gallagher family today, Board of Advice members, other distinguished guests, ladies and gentlemen. May I welcome you all to an event that I consider very special to the Institute, and that's the Peter Gallagher Memorial Glycomics Lecture. It's a very special event because Peter Gallagher was very special. He was a long-term supporter of the Institute, and before that, he was a legendary rugby league football player, as you can see in this wonderful diagram, an Australian representative and indeed a captain of our national team, the Kangaroos. Peter was also an adored member of the Gold Coast community and did an enormous amount of work in the Gold Coast community, particularly in his roles within the racehorse industry. He was very much a can-do person, and that is certainly what he brought to our Board of Advice that many years ago before he passed away. So we celebrate Peter's life every year for his contributions that he's made, not just to the Institute, but also to the Gold Coast and to Australia in the broadest context, by hosting this special public lecture in honour of his life. So moving to the lecture today, and by way of introduction, our 2014 lecturer is indeed a distinguished glycoscientist, researcher from Imperial College London. It's my pleasure to welcome Professor Anne Dell, who is, by the way, even though based at Imperial College, and she knows I was going to say this, is an expat Western Australian. In fact, she graduated from the University of Western Australia in chemistry at that point in time, and then left Australia in the early 70s to advance her research opportunities, taking on a higher degree at the University of Cambridge, supported by an 1851 exhibition research scholarship. And for those who don't know, that's a very prestigious scholarship award that enabled particularly Australians to head to the UK to undertake higher degree research. After completion of her PhD, DPhil probably is the more appropriate title. No, PhD?
DPhil is Oxford.
DPhil is Oxford. Sorry, I got it wrong. And then moved to Imperial College to commence her career, where she quickly rose through the ranks to professor in 1991. I could go on and on and on about Anne's career trajectory, but I won't. Just to summarise it, Anne, at least I think in our community and indeed in the scientific community, is one of the world's leading outstanding research leaders in the area of carbohydrate chemical biology, and has made enormous contributions to this important research field. Importantly, Anne is indeed a marvellous icon for all students, and for those students particularly who aspire to great things, particularly female students. Anne achieved all of her outstanding contributions with time for a family, and I think that's such a wonderful thing that we see, that you don't have to choose one or the other, you can actually do both, and Anne's such a wonderful icon that I think all of our students can look up to. In recognition of her research outcomes and achievements, she has been bestowed numerous awards, including appointment as a Commander of the Order of the British Empire, importantly elected to the Fellowship of the Royal Society, and awarded a Wellcome Trust Senior Investigator Fellowship. All of these are signals of a person that has done amazing things in their scientific career. So we're delighted to have her. I have no doubt that you're going to enjoy her lecture entitled Communication with Sugars. Anne, the floor is yours.
Thank you. Does my microphone work now? It's brilliant, isn't it? It worked over there, now it works over here. So Thank you, Mark. It's wonderful to be here. Thank you very much for the invitation. Thank you very much to the audience for coming, I think, some distance, some of you, in traffic to hear all about communicating with sugars. So let me see whether this is going to work. Yes. So having been told that we were celebrating Peter Gallagher, I thought, great, I can say I know all about rugby because I actually live a couple of miles away from Twickenham Rugby Stadium. And the supermarket I normally go to is here. That giant big car park is usually used as an overflow car park when the Twickenham car parks, which are always full, are filled up, and which means I can't go to the supermarket. So when there's a big match on, I complain. However, having prepared this slide nicely, one of my colleagues at work said to me, is Peter Gallagher a rugby union player? And I said, what do you mean, rugby union player? Rugby's rugby, isn't it? So a couple of hours later, I was by then an absolute expert on everything to do with the difference between rugby union and rugby league. But I thought I would continue to use my slide anyway. I am now sufficiently expert that I know that the grand final, or whatever it's called here, of rugby league was played only a couple of days ago, weekend or whatever, with the Rabbitohs winning. And I also know that the reason that the Rabbitohs won is because they had 3 English players playing for them. 3 brothers, with one of them winning the man of the match, yes? Yes? Right, so, okay, so let me make sure I can work out which key works. So communicating with sugars. Now, I do appreciate the audience is quite diverse, and many of you will feel you don't know anything about sugars, but actually all of you do. I don't think anyone in the audience will be unaware of glucose, particularly with all the publicity given to type 2 diabetes and the need to have the right levels of blood glucose. So glucose is a sugar. It is, if you like, the most famous sugar ever. We wouldn't be here on the Earth if glucose wasn't around. It is, as far as molecules are concerned, molecules containing carbon, which are organic molecules, it is the most abundant organic molecule on the planet. And it is largely that abundant because of cellulose in trees and other plants. So cellulose is a polymer of glucose. Oops. The glucose, as well as it being part of all of the plant world, it is the most important energy source for everything, including ourselves. But I'm not planning actually to talk very much about glucose today. I want to talk about relatives of glucose. So glucose is a relatively small molecule with 6 carbons. And there are a lot of other related sugars that actually look very similar. And I don't expect you to remember or even think about the structures on here. Most of the time, for the rest of the talk, I'll be using these little cartoon symbols. So all you have to remember are a few colors and a few shapes. And what this slide is indicating is that the other sugars that I'm talking about can be derived, and usually are, from glucose, and are actually very, very similar to glucose. So for example, the sugars which are being shown as circles of different colors, if your eyesight is good enough, without my glasses, mine isn't, I can't see you and the slides at the same time, which is why I'm holding my glasses in my hand. The colors are indicating what we call stereochemistry, or in other words, shape. And the difference, for example, between the blue color here, which is glucose, and the yellow color, which for some reason this keeps shimmering, and I don't know what I'm doing to do that, galactose, there is an oxygen which on this sugar is pointing up, and on this sugar is pointing sideways. And that's the only difference. It's just whether it's up or whether it's sideways. And that makes it a different sugar. It's called galactose. And similarly, mannose over here is in this case, you can spot the difference. It's actually this oxygen is pointing in the other direction. I'm going to tell you a lot about mass spectrometry today. You might think mass spectrometry is something either you've never heard of, or if you have, it's not very interesting. What I'm hoping to tell you is that it is interesting. What a mass spectrometer does, as we'll see in a minute, is to measure mass. And with respect to these colors, the only thing that you have to remember, you don't even have to remember that, but it will help you to understand some of the slides, is to remember that anything which is the same shape has the same mass. The colors indicate also a relationship, but you don't need to worry too much about that. But if they have a different shape, they are actually a different sugar. And you'll notice this one has a nitrogen here instead of an oxygen. But that's all you need to understand about this slide. And I promise you that after this, hopefully things will get a little more interesting. So Mark has already said that I am at Imperial College. I thought it might be nice if you haven't had the chance to go to London, therefore you will not have had the opportunity to visit this part of London, which is Hyde Park, Kensington Gardens, and the most important bit, which is Imperial College. And Imperial College sits in the middle of this rectangle here. Up here is the Royal Albert Hall, and down here are a variety of museums, natural history, Science Museum, and over here the Victoria and Albert Museum. And Mark has already given quite a nice introduction to what I plan to talk about at this point, because he mentioned the 1851 award, which I went to the UK on. And this dates back, 1851 is the year, to 1851, and it dates back to the first Great Exhibition, as it was called, but the first of the Science World Fairs. So the UK held the Great Exhibition in 1851 in Hyde Park, and the amazing thing about this exhibition was that it was housed in this so-called Crystal Palace, which was a largely plate glass building of sort of cast iron and plate glass. And the interesting thing about it is that the ability to make plate glass had only actually been worked out just a few years before 1851. So the modern technology was being exploited. The other thing, particularly for the younger people in the audience, as to how far you can go very rapidly, is that the design of this building came from a person called Joseph Paxton, who had been a gardener and was actually a head gardener for the Duke of Devonshire. And Joseph Paxton was the youngest son of a farmer. At 15 years old, he was apprenticed as a gardener and went to work for the Royal Horticultural Society in the UK. So he had, apart from farming training, he had virtually no training, but he was a bright lad and he did well. And the Duke of Devonshire used to visit the RHS gardens in London and used to chat to this young gardener. And when he was about 20, I think, the Duke of Devonshire mentioned that because he knew so much about gardening, perhaps he'd like to be head gardener for him at Chatsworth House. And no one really knows historically whether this was a serious invitation or not. But within days, Joseph Paxton had packed his belongings and headed to Chatsworth. The Duke wasn't there at that point. He'd gone traveling in Europe. He had got to know the head housekeeper and chatted with her, got to know his future wife, who was one of the servants, and essentially started to rebuild the gardens of the Duke of Devonshire. And he, although he was not a trained architect, he drew a sketch for the commissioners for the 1851 of what he thought would be a nice house, as it were, home for the Great Exhibition, and it was accepted, and within 9 months it was built. So this is a remarkable achievement for engineering. It also is a remarkable achievement for public outreach, because the exhibition was open from May of 1851 to October, and in that time, 1/3 of the population of Britain attended. And bearing in mind that the railways had only pretty much just started at that point and people had to get to London. So something like 6 million people went to the Great Exhibition in that time. And the Great Exhibition made a profit, a very handsome profit. And from the money of the Great Exhibition, all of the land here, all of that land, and in fact land all the way out here, which was subsequently sold, was purchased by the Royal Commission. And in addition to that, as Mark has already mentioned, they used profits to set up an educational trust to enable students from the colonies, of course as they were then, to study in the UK. And I was very fortunate to be granted one of these, and I thought, especially for the younger people in the audience, you might like to see what was the email of the 1970s. So this was the notification that I had one. And I was very fortunate because I was able to go to Cambridge to study for my PhD, but I was even more fortunate because although I did not go to do my PhD with Howard Morris, I, for a variety of reasons, finished up being his very first PhD student in his very first academic appointment in Cambridge, and became a PhD student studying mass spectrometry. I just want to point out here, this was sort of the pot of gold at the end of the rainbow, was the opportunity to be a student at King's College in Cambridge, which is, as you may know, the Cambridge and Oxford students join colleges. I chose King's College because I thought it was a very traditional college and thought that would be nice to go and stay at a traditional college. Again, a complete mistake, but turned out to be a really good mistake because it suited me very well. King's College was actually incredibly left-wing. Keynes, the famous economist, had been the bursar of King's College and had actually built it up very financially successfully, but also very much created the culture. E.M. Forster was a fellow, had lived in residence there. So it was the kind of college which attracted a great variety of students from countries such as the Soviet Union. And the first year I was there, I remember in the summertime, we had visitors from countries like the Soviet Union staying at the college. And it was, for me, as a parochial West Australian, meeting these people for the first time, it was a tremendously stimulating environment. So mass spectrometry, a little bit about what it is so that you'll have some idea of what I'm talking about. Essentially, it is a fancy weighing machine. It allows you to measure the mass or the weight of, in this case, molecules very accurately. And it's very, very widely used. And in fact, I'm sure many of you will be familiar with some of its applications. For example, the testing for performance-enhancing drugs is almost entirely done by mass spectrometry these days. Fuel testing for the Formula One races. If you watch a Formula One race, you will see just before the race starts, a guy running from one car to the other. What they're doing is sampling the fuel, and that is, within 24 hours, is analyzed by mass spec to make sure that the right fuel is being used. Forensic analysis, analysis of chemicals, but for the point of view of today's talk, the analysis of biopolymers, and associated with that, the modern biologic drugs. Again, some of you will be familiar with the biologic drugs like, although the names might be different here, Herceptin, for example, the anti-breast cancer drug, which is widely used in the UK and the rest of the world, is a biologic drug, and what that means is it's a biopolymer. So I was very lucky because I started my career in mass spec at a time when mass spectrometry, for the very first time, was being applied to biological molecules. And the reason it could be applied to biological molecules was largely because of ideas of my mentor, Howard Morris, who had come up with ways, at least in his head, and then industry responded by building these things, of being able to analyze very large molecules. And a biopolymer is simply a polymerized pieces of oligomers, as they're called, or individual sugars, for example, will polymerize together to make a bigger molecule. And for mass spectrometry to work on these larger molecules, you needed instrumentation, which at that time was essentially very, very powerful magnets. And these actually had not been built until then. So theoretically, they could be made. Practically, they had not been built. And the very first ones, the first one is this one here, the second is this one here, these were built and delivered to us at Imperial College. And at that time, nobody else had them. So that was brilliant. So I was a postdoc by then at Imperial College. And people came from all over the world to us, asking us if we could help them with their generally their biological problems. And what I'm hoping now to do is to give you an idea of the kinds of things that we do and how that information that we get from our research can be used to understand biology better, but also, after that, to translate it into new medicines and generally better healthy lifestyles for people. So what is mass spectrometry? As I said, it's a fancy weighing machine. What we do in mass spectrometry is we take a sample of any kind. So that, let's imagine this is a biopolymer with the individual units joined together. It's a very happy one, which is why it's got a smiley face. And its tentacles, as it were, antennae, are kind of droopy. And what happens is we ionize it. And that simply means that we take that molecule and we give it a charge. It has to have a charge because otherwise you can't measure its mass. And there are many ways of giving a molecule a charge. And back when I started, one way of doing it was to take an atom or ion gun and effectively fire accelerated atoms or ions at the molecule. And this, again, was a new technology. It was developed in the UK in Manchester by Mickey Barber and colleagues at the University of Manchester Institute of Science and Technology. And all of this happened pretty much at the same time. The development of those very big high field, as they were called, magnets, and this really nice technology for ionizing our samples, allowing us to get mass spectra, as we call them. And what are mass spectra? They are simply, it's data or information that tells us here the mass of the molecule and these other components here. Notice that I've changed the color as I converted it to a charge. And also, as you can see, its face now is no longer happy and smiling. It is actually looking, ouch, I am very hot. And the heat, if you like, that's put into the molecule in doing that is enough to make it vibrate and fall apart into pieces. So we get in what's called the mass spectrum, which is just simply an axis along here with ions of different mass value, which tell us about the mass of the overall molecule and the masses of bits. And what you'll notice here is that we can differentiate between the ends of the molecule, because this one's got the, now rather scared, face on it, and this, for example, is the tail. So we can piece our molecule together. So it's a bit like thinking about a jigsaw puzzle, that we've dropped the puzzle on the floor, it's all in bits, but not entirely completely smashed up, and then we try to reassemble it and work out what it was in the first place. So that's mass spec. Why are we interested in sugars? Well, the main reason we're interested, well, there are many reasons we're interested in sugars, but one of the main reasons we're interested in sugars can be exemplified by just thinking about biology and what it is in biology that essentially codes for a biological system. Now, the first thing that most people, even I would imagine most of you, if you're not, or many of you who are not scientists, will think about is the DNA, because genes and DNA is what people often know about. But about 15 or so years ago, when, or slightly less than that, when the Human Genome Project, the outcome of that was announced by the President of the United States and the Prime Minister of Britain, that's how high profile it was realized to a lot of people's dismay that the number of genes in humans, despite earlier predictions that it ought to be at least 10 or even more times greater than this, was very little different from the number of genes in this, which is a very, very tiny worm, which is about 1 millimeter long, this, which is the fruit fly, and this, which is a weed. It's a little cress weed, which, at least in England, you get in car parks growing, and it's used as a model organism in the lab. So it's rather pathetic. It has little tiny flowers, but that's about all. It doesn't do anything special. But notice it has more genes than we have. The other thing that is also, or was also discovered, but again, perhaps not quite so surprising, was that the chimpanzee and the human actually not only have exactly the same number of genes, but actually the sequences are 99.9% identical. So biological complexity is not linearly related to the number of genes. And there are many reasons, which if I'm sure there are many in the audience who will say, what about, for all the other reasons why why we think we are more complex than, for example, a 1 millimeter worm. But one of the most important reasons, and for this audience, the most important reason, is because the products of the genes, so the genes encode information that leads to proteins. So DNA goes to RNA, goes to proteins, is the paradigm of life. But the proteins do not stay as proteins. They become modified. And the greatest diversity for modification is putting strings of sugars on them. And what we get from that are what are called glycoproteins, or proteins that have sugars on. So the functional diversity of the gene products is vastly amplified by having the sugars on them. Now, whereas we can sequence the DNA and know exactly what the DNA sequences are, and of course, nowadays that has become much faster and much cheaper, knowing what these glycans are is still a very difficult thing to do. And so today I want to give you a glimpse into the research that's been going on now for 30 years or more, but also I hope by the end of my talk to give you an idea of actually what still needs to be done. In other words, we do know quite a bit, but there is so much we don't yet know. What we do know is the following, that every single cell in every organism that's looked at is coated with sugars. Now, this is a picture that's called an electron micrograph, and essentially what it is, it's a slice through a cell, and then an electron microscope, because the cells are tiny, is used to visualize that slice. And on the surface of the cell here, there is a dye which has been used. It's called ruthenium red, which has been used. It's shown in black here because it's an electron microscope picture. The black fuzzy bit around the edge of the cell here, this is a portion of the cell. This is the nucleus, a portion of the nucleus, and this is what's called the cytoplasm or cytosol. You'll notice that the whole of the cell surface is covered in this black coat. The dye is recognizing sugar. So what the dye is lighting up, as it were, even if it's lighting it up in black, is the sugar coat. And every single cell in our body is coated with sugar. So, you know, we are sugar-coated people. And these sugars are linked on. So this is the membrane. Don't worry about the details of this. This is the membrane, or the outer surface of the cell. And the sugars, which are shown by the symbols that I've shown you already, are linked on as these sugar polymers onto proteins, which are shown in these different colors here, blues and brown. And they're also on lipids, but we're not going to talk about lipids today. So as I've said, they are on our proteins. And we have very, very good evidence that's been built up now over 2 or 3 decades that these glycans are involved in recognition processes. So for example, these are cartoons of cells cut out just so you can tell it's a cell, showing there's all sorts of organelles inside. But what we're showing here is that when we have a cell from, for example, a mammal, perhaps a human, then when a pathogen, an infective agent like an infective bacteria or a virus, and of course influenza is something that obviously this institute is very famous for, when the virus is infecting the cell, it will engage with a sugar on the surface of the cell, and what it has is a very specific protein called a lectin on its surface that recognizes that sugar. Similarly, if you have, for example, this is a E. coli pathogen, which is a urinary tract pathogen, is recognizing sugars. When a mammalian cell meets another mammalian cell, exactly the same recognition is taking place. Families of lectins, families of sugars, which they recognize. And so, for example, if we're looking at these are white blood cells, so this is a picture taken when a camera has been put up a capillary. This is a white blood cell, and the reddish color that you can see here is the layer of cells that lines the inside of your capillaries. And the white blood cell engaging with the endothelial cell, as it's called, that recognition involves sugars and lectins. And what our mass spectrometry can do is it can tell us what kind of sugars are on these cells, because we're trying to understand these recognition processes. And to do that, we need to know what the sugars are. We need to know what the recognizing lectins are. So one of my very early achievements, which is something which at the time we had no idea of the importance, but actually it's taken a decade or more, and even now we don't fully understand the importance, was the discovery of, because of a collaboration we were carrying out with a scientist who'd come to us from Southern California, from a cancer institute in Southern California. And he was interested in how leukemia cells differed from normal white blood cells. Just generally, at that time, just wanting to know the fundamental biology and obviously subsequently the potential exploitation of that for diagnostics and possibly anti-cancer drugs. So he asked us whether we might be able to tell him what kind of sugars were on the surface of white blood cells, and we said we'd give it a go. And one of the sugar clusters we discovered was this little cluster here, which, again, I've put the symbols on so that you can see the sugars we were looking at earlier. And this is a very, very famous sugar cluster now in glycobiology. Generally, it is called Sialyl-Lewis X. And this arrangement of having one of these diamond sugars, it's a very important sugar. This is the sialic acid. In fact, that's what the flu virus recognizes. This is galactose, so-called glutnac, and fucase. So this little cluster we're going to see in a number of later slides. Now, what we identified at that time, which subsequently became very important, we observed that in leukemia cells, they had a lot more of this Sialyl-Lewis X structure. About 5 years later, the partner, the lectin partner of Sialyl-Lewis X was discovered. It turns out to be a family, so there's a family of 3 members of the family that will engage with Sialyl-Lewis X and recognize it. And these lectins are actually called the selectins. And what was worked out, again over a number of years with quite a lot of research labs around the world being involved, is that the way that a white blood cell, like for example a neutrophil is one of our white blood cells whose job it is to kill nasty things if we get infected. So normally it's circulating in the blood. If we get an infection in our tissues, an inflammation occurs, then the neutrophils have to know it's there. They have to recognize that they need to go out of so this side, this is the endothelial layer, which in that proper photo was red, or pinky red, now coded green, just to confuse you. So we have this layer of endothelial cells, as they're called. This side of it is the capillary, this side is the tissue. So what the white blood cells have to do is they have to get from that side to this side. And of course, they're moving. If you think about it, they're being pumped by the heart. It's flowing along here. What they have to do is that they have to come to a stop, and then they have to squeeze between the cells that are lining the capillary and get to the other side. And medical people call this extravasation, getting to the other side. So this is a cartoon showing what's happening, that we have our Sialyl-Lewis X on our white blood cell. It is engaging with the selectin, which is being expressed on the endothelial cells. And it's a bit like rolling over Velcro. If you think about when you have a ball and you attach it to Velcro and then try to move it along, it will obviously, it very rapidly comes to a stop. Then it flattens and squeezes out. And this engagement of white blood cells with the endothelial layer absolutely underpins everything to do with white blood cell trafficking in the human body. So, for example, as I'm sure you all know, you have 2 circulatory systems. You've got the blood and you've got the lymphatics. And the lymph nodes are the really important connections where things from the blood can go into the lymphatic system. And some of the white blood cells have to do that. For example, there are white blood cells called lymphocytes that need to go into the lymphatic system in order to circulate and do their job as immune cells. Some of them, like the neutrophils, do not. They have to stay in the blood system until there is some kind of infection out in the tissue. So again, there has to be regulation at the lymph nodes defining when these white blood cells and which of them are allowed to leave the endothelial layer. And that's why the discovery that Sialyl-Lewis X on cancer cells is much more highly expressed was a very important observation that was rapidly proved by a whole variety of other methods in the 1980s, where it was shown that pretty much all solid tumors, especially those that metastasize, i.e. migrate to other parts of the body, were expressing high levels of SALR Lewis X and almost certainly use this trafficking pathway to get from the site of the primary tumor. So what I'd like to do now is to tell you a little more about how we can use, so that was the 1980s, but as you'll see in a minute, we are still looking at those kinds of structures. But I want to give you a better idea of how we can use our data in a wider spectrum of applications. And this is just to point out what we are doing overall. What we're trying to address is what kind of glycans are we making in particular in the human, because we're trying to understand human biology. We very often use the mouse as a model system for the human, because obviously it can essentially reproduces faster for a start, and it is clearly ethically more appropriate to be doing experiments on a mouse than it is on a human. We also will look at cells, and in fact we can look at a whole variety of biological substances. But the aim is to get pools of glycans that are representative of the material we had in the first place to address the question of how might these glycans be involved in recognition processes that lead to biology. And this is one of our students in London with one of our modern mass spec systems. Now, I'm going to have to give you a little bit of biochemistry knowledge, otherwise you're not going to understand my cartoons. But don't worry, it's very simple, or at least I think it'll be simple. So basically, what you need to know is that inside a cell, we have a production line for making glycoproteins. So the proteins themselves, so this is the nucleus of a cell. Again, it's only a partial cell. So this is that plasma membrane with the sugar coat is not being shown on this cartoon. But inside the cell, we have what is called an endoplasmic reticulum. Don't worry about it. Everyone calls it an ER because they find it a bit hard to say, endoplasmic reticulum. And this is where the protein is first made. So this is where the information from the DNA in the nucleus is translated, essentially the DNA is converted to RNA, and then that information is translated to a protein sequence, which occurs here. And then a glycan, a sugar polymer, is put on it. Now, what is absolutely amazing about that, and this is our glycan polymer with the cartoons that I showed you earlier, but what's absolutely amazing is it doesn't matter whether it's a human or a yeast or a plant or any other animal which is what's called a eukaryote, in other words, an animal which has a nucleus. This glycan is absolutely conserved. This has been conserved since the beginning of evolution, as it were, which I think, and many people in glycobiology think, is just an absolutely amazing conservation, but it reflects almost certainly the importance of this particular modification. So absolutely conserved. What then happens is that in the ER and then subsequently in another apparatus within the cell called the Golgi, there is a production line where this is trimmed down and then built back up again. So I'm just going to show you what happens. It gets trimmed and trimmed and trimmed further, but then it starts to get built back up again, and then it finishes up on the surface. And the marvelous thing about this is that all of the glycans have a common, what we call core, as they're attached onto the protein, because it never trims right down to take the core off. So we know what's in the core. But the other thing that's important is that we get what are called antennae, and those antennae are decorated, and you already know one decoration, that's here, SALR Lewis X. Arrange it with exactly the same set of individual units, but in a different arrangement, we get a related sugar cluster called SALR Lewis A. Take off the sialic acid, and we get Lewis X. Lewis X is on all of your white blood cells. SALR Lewis X is on most of your white blood cells. You will have heard of the ABO blood group system. Those are sugars. So basically, again, you'll see that they're all sort of related to each other. So they're just little patterns of sugars. And these sugars are there to, as it were, identify the surface of the cells. So what do we use our mass spec for? Well, we use our mass spec, as I said earlier, to get a mass or complete weight, and then the pieces. So this is modern mass spectrometry, where I had guns earlier on. Unfortunately, we don't use guns anymore. It's not quite as exciting. What we use instead are lasers, like a very high-power laser pointer. That's what the L in this acronym stands for. Don't worry about the rest of the letters. Or we spray the sample into the mass spec, which is what that other acronym is. So it's just a different way of making the ions. Happens to be more sensitive than the ways we used 20, 30 years ago. But we get our molecular ions, and then, as I said, we get our fragment ions. The difference between now and 30-odd years ago is we do these in 2 experiments, as it were. We do it in 2 stages in the mass spectrometer. So we can do this stage just by itself, just getting the masses, or we can choose to do this as well. So what does that mean? Because you now are absolute experts in the kind of glycans that can be made by that biosynthetic pathway, you will hopefully not find it too difficult to understand that if we have a mass which we can fit to a composition, and don't worry about the fact that this is not the words I've been using, hexose just means that we can't tell the difference between a green circle and a blue circle and a yellow circle because we said they were the same mass. So we can get the composition in terms of these, what we call isomers. But the important thing is we can immediately map it onto potential structures. So we use that knowledge of the biosynthesis to do that. And if we then want to know what structures they are, we do MSMS, where it falls apart and gives us beautiful ions that tell us exactly where our decorating sugars are. So what that means is that we can take a range of white blood cells, and these are examples. I've already mentioned a neutrophil. There's another cell called a mast cell. There's another cell called an eosinophil. These are cells that are really important in our immune system for helping to keep us healthy. And all that I want you to look at is not any of the detail. It's not intended to show you the detail, but just to show you that we can take the whole cell and we can get this from the mass spectrum, these populations of glycans, and we can attribute structures because we know these biosynthetic pathways. So how can we use that information in a real-life context? And I want to talk about 2, briefly one, and then a little bit more time on the second example. So I want to firstly tell you about the fact that there are, and this has been known for some time, a number of diseases which are called congenital disorders of glycosylation. And all that means is that congenital, of course, means born with, so there's a problem already at birth. Disorders of glycosylation means something went wrong with those glycosylation pathways. And many of these are actually, unfortunately, really devastating diseases because, as I said, these recognition events are so important, they're important in early development, that if something goes wrong, the chances are that that's going to cause a lot of problems. So many of the really severe congenital disorders of glycosylation have really been known for some time because usually children do not survive very long if they have them. And there are actually screening programs, and now that we understand the glycosylation, there are screening programs around the world for identifying children who are born with these diseases and finding out very rapidly what they are. However, it's become very clear in recent years that there are less severe congenital diseases of glycosylation which people have not understood, and understanding them better can obviously lead to better therapy and hopefully a better quality of life for the children involved. So I'm going to give you one example of that, and then I'm going to spend a little bit of time, I'm looking at the clock and trying to remember when we started, to tell you about recognition in human reproduction, because recognition and the correct recognition is incredibly important for many aspects of human reproduction. So let's start first of all with the CDGs, and I'm afraid there's a few sort of medical and slightly complicated acronyms, but I will explain them and hopefully it won't be too complicated and that you'll understand the theme of what I want to say. The first thing, however, I want to say is that all of modern science is done with teams of people rather than single individuals. And one of the great things about not even so much modern science, even when I first started, was that science is very international and very often involves expertise from a range of places. So in this particular case, we were carrying out the work in collaboration with Christoph Klein in Munich, and the patients that he was working with were children in Germany. And in London, at University College London, linked to Great Ormond Street Hospital, which is the main children's hospital in London, Professor Tony Siegel and a PhD student, actually a trained doctor, but had decided to go and get a PhD after having had medical training, Boo Hussein, was involved in the project with Tony Siegel, and in our own group, Aristotelis Antonopoulos, a postdoc, did all of the lovely Glycomics work that I'll show you in a minute. Now, what Christoph Klein discovered around about 6 years ago, 2008, 2009, is that a type of dysfunction, which is not uncommon in children, which is called severe congenital neutropenia. What this means, again, congenital, you know what that means, severe. Neutropenia simply means there's not enough neutrophils being made. So there are a lot of instances of so-called SCNs, and very often having an SCN is not a major issue clinically, because it often just simply manifests itself as problems, for example, with infections. So these children and then young adults, they do have neutrophils, they just don't have as many as a normal healthy person has. So they are prone to infections, and obviously they can be treated for that. But there are some SCNs where there are other phenotypes or other clinical symptoms. And the one that was being looked at in particular were families where the children with severe congenital neutropenia had other physical defects such as short stature. They also had heart problems. And importantly as well, not only did they not have as many neutrophils as they should, those neutrophils didn't actually work very well. Now remember, the neutrophils are the white blood cells that go out into the tissues to kill things. And the way they kill things is by spewing out chemicals. They do other things as well, but their main thing is to spew out chemicals. And actually what they do is they make hydrogen peroxide and related sort of high-energy oxygen intermediates. And so these children were actually, their neutrophils were not producing the hydrogen peroxide they should. And this is shown on this slide, which I apologize, I've had to put a structure on for you, that what Christoph Klein had discovered was that the gene that was mutated in these children was the gene that coded for an enzyme, which is called G6PC3. It's written out up there, but what all you need to know is it's important in glucose metabolism. What it does actually is to take a phosphate off what is called glucose-6-phosphate. So glucose metabolism is being affected by the problems in this gene. This little bit of data here, I haven't put much data on the slides, but a little bit of data here is just showing the neutrophil dysfunction. So what we have here are 3 children called A, B, and C from 3 families. Child A, a boy, actually, I shouldn't call him a child because he's now about 18 years old, is the brother and actually twin brother of what is called healthy control here. So the healthy control, these are not identical twins. The healthy control, in terms of age matching and obviously sort of environment, is a perfect control for this child. And the other children are shown here. And the height of this bar graph is just showing how much superoxide is being produced by their neutrophils, by each million neutrophils. We have lots of neutrophils. So 1 million neutrophils is producing that amount of hydrogen peroxide in the case of a healthy control. And as you can see, the other children, particularly child A, has a very low level of hydrogen peroxide, showing this neutrophil dysfunction. So what we were asked by Tony Siegel, who at University College had identified families in London whose children had this same defect. So there were families in Germany and families in London, and also now known to be families elsewhere. We were asked, could we explain how a defect in glucose metabolism was influencing how the neutrophils were functioning? And one of the questions we decided to ask was the neutrophil able to make its normal glycan population? And what we're showing up here, so this is the healthy twin, and this is a mass spectrum of the neutrophils taken from that healthy twin. And again, all you need to look at is just simply an idea of the patterns, showing, as you can see here, that we have lots of different glycans. They have those patterns of antenna that I showed you earlier. And what was really dramatic in his twin brother was the complete absence of all of these multi-antennary glycans. So the conclusion from that, if you just simply look at this picture here, was that this is what we expect on normal neutrophils. Lots of antennae, lots of these decorating sugars, those diamonds and red triangles, which are fucose and sialic acid, for anyone who wants to know the sugars. But this is what was happening essentially in this, not only in the healthy twin, the affected twin, but also in the children in the other families. And so it was immediately obvious to us what the problem was, and I think it's probably obvious to you. The yellows are not going on, because if you don't get the yellow going on to make the first bit of this, it can't put anything else on. And the yellow is galactose. And this, for anyone who knows about biosynthetic pathways, is just simply showing the complicated interrelationships of molecules inside the cell. The important thing to note is that glucose here is feeding into what is called here UDP-Gal, which is essentially galactose in an activated state, ready to go onto a glycan on a protein. So this is a very important observation because it immediately suggests that there might be possible to improve the clinical symptoms of these children. And in particular, it might even be, although we don't yet know, we are hoping that it might even be possible to do that just by giving galactose supplements in the diet. And this is currently under investigation in Germany. The child being treated in Germany after 6 months does appear to be better, but it's early days. I don't mean better as in able to be perfectly normal. I mean the symptoms not as severe in terms of infections and so on. But it is very early days, but this is the way that we can take basic science through to translation. So the second example I'd like to tell you about, in a little bit more detail, but not too long, because I notice time is getting on, is to talk about human reproduction, and in particular to address the question of how it is in human reproduction, and in the reproduction, in fact, in all mammals that essentially have the developing embryo in an environment where the embryo is foreign, as we'll see, to the mother or the female animal that is carrying that embryo, this issue of self and non-self. Because the whole of the immune system's function depends on it being able to recognize what it knows as self and what it recognizes as non-self. And it is programmed to attack non-self and to not attack self. And of course, as I'm sure many of you know, if that programming goes wrong in some way, in, for example, autoimmune diseases, then the immune system will be starting to attack self when it shouldn't. Now, a lot of what I want to tell you about today has been done with our collaborator, Gary Clark, who's now at the University of Missouri. And we started working with Gary in the early 1990s at a time when I knew nothing about any of this. I was trained as a chemist. I've learned some biology along the way. It's sort of paper thin at times. And one of the things that I hadn't really thought about at that time were some fundamental questions that had actually been posed a long time ago, like this question here that had been posed by a very famous British immunologist, Peter Medawar. And he asked, way back in the early 1950s, this question: how does the pregnant mother nourish within itself for many weeks or months a fetus that is antigenically a foreign body? Leaving aside the fact that he calls the mother an it, rather than... this is a very important question, because the key thing to remember is that as soon as the embryo starts to go from the single cell stage when it was, if you like, the mother's egg, it is expressing foreign things because half of its DNA comes from the father. Now, there are other questions which are related, which are, for example, and again, this is slightly complicated, but don't worry about the cartoon, is that the way that the immune system recognizes that our cells, our self cells, the immune system shouldn't attack those cells is because on most of our cells, we have these proteins, or actually glycoproteins, that are called human leukocyte antigens, or HLA. And they're essentially the molecules that say, please do not kill me, do not kill the cell, I belong to you. Now, the thing about both of the gametes, the egg and the sperm in the human, is that they actually do not have these HLA antigens. In addition to that, when the embryo implants into the womb, and this is the placenta here, this square here is now blown up to show here. What the embryo does, as the placenta, which of course comes from the embryo, therefore has father's genes in it, these cells start to infiltrate in order to connect with the mother's blood system. And so we have a whole variety of cells with all fancy names. Even I can't pronounce them, syncytiotrophoblasts and stuff, don't worry about it. We have all of these cells that are going to engage, some of them will engage with the mother's cells. They also do not have these HLA antigens. So again, in principle, they're foreign. And in particular, there is a class of white blood cells that I haven't mentioned up to now. There are lots of different kinds of white blood cells, which have a really interesting name. They're called natural killer cells. Their job is to kill cells that lack HLA antigens. So they go around looking for cells that don't have HLA antigens, and they kill them. So the really important question, which I have to say, I'm not going to give you the answer because this is research that's on the go. We're a little way to understanding it, is how do women, for example, protect the developing embryo? How do they essentially protect sperm when they enter women, because they don't go and kill off all the sperm, when natural killer cells are in effect trained to kill these cells that lack these antigens, but they don't? And the important thing is, you might say, well, maybe there's no natural killer cells in the human reproductive tract. In fact, about 70% immune effector just simply means immune cells that are active. In the human uterus, about 70% of them are NK cells. So we don't actually know the answer to this as yet. There are all sorts of experiments, and it's not going to be a simple answer either. There are going to be a lot of different factors that are involved. But it almost certainly that a very key factor involved is recognition, and glycans will be involved in that. And so Gary Clark, the person I showed you earlier, back in the mid to late 1990s, when he was thinking about this, he suggested, and he called this, again, a fancy term, feto-embryonic defense system, or human FEDS. He suggested that essentially, that if we have HLA negative molecules, they have to have something else on them that say to the natural killer cells, please don't kill me, I'm actually friendly, I am not foreign. And he suggested that there were sugar sequences. And another very important thing he suggested at that time was that the sugar sequences that are on the human egg may be important for two roles, because remember, the human egg then becomes the embryo, that they will be involved in recognition of the sperm with the egg, because when you begin the process of fertilization, the sperm has to recognize the egg. You don't want the sperm recognizing a neutrophil or a T cell or any other random cell it happens to encounter. And he was proposing that the sugars that are on the coat, it's called the zona pellucida of the human egg, may perform a dual role of being involved in sperm binding, but also to help to protect the gametes from immune attack. And it took some time to get evidence for that. And I'm very pleased to say that once we were able to get evidence of what is on the human egg, and the teams of people again involved here from a number of countries, we found a little bit to our surprise, because we didn't think it was going to be quite that easy, that the major glycan that is on the surface of the human egg is this epitope that we have already identified earlier on in the talk as being very important with respect to the functioning of the immune system, SALA Lewis X. And what was very surprising to us, and I thought I'd put a bit of data just so that you can see some data, was that when we did our profiling, that SALA Lewis X was pretty much on everything. And we were also very pleased that this kind of work was considered important enough, because as scientists, what we like is to have our papers A, published, secondly, published in the very top journals, and thirdly, noticed by the editors so that they make comments about it. And this we were pleased happened in this case, especially because they thought it would be a nice story. So it was called The Sweet Meeting of Sperm and Egg, and it started off, first your parents had to meet, but another, etc., etc. And of course, that was the paper where we were reporting that the egg is covered with these sugars. However, if I am allowed another 5 minutes or so, yes, I don't want you to leave you with the fact, or that you might be thinking at this point, that science is all very straightforward. Research gives you the answer you wanted in the first place. You have your hypothesis. It's giving you the right answer. Therefore, you know everything. Because it turns out that it's not quite as simple as that, and we know that we don't know everything, because one of the things that we had known for a decade before the discovery of SALA Lewis X on the egg is that surrounding the baby as it's developing, the liquid, the so-called amniotic fluid that bathes the embryo during development, has, during the time of the development of the embryo, a glycoprotein called glycodelin A, or GDA, which is at very high abundance in that fluid and is known to be really, really important for helping to protect the developing embryo. And what was also known about glycodelin A, using the technology that's used in vitro fertilization, is that glycodelin A was able to block sperm-egg binding. So in other words, somehow it is able to mimic one or other of the molecules that are involved in that binding. And so we had done the characterization of glycodelin A, and regrettably, it has absolutely no SALA Lewis X on it. So in other words, it is possible to take a protein which has a range of sugars on it and block that interaction, even though those sugars are not SALA Lewis X. So we don't yet know the whole story. However, what we do know is that glycodelin A is there's a lot of words on here, don't worry about the detail of it, but glycodelin A, as I've mentioned, is very important in protecting that fetus during development. And what we also know, and that's why I've shown you a complicated slide slightly faded out so you can't see it, is that it has lots and lots of different glycans on it, and they are summarized down here. We know that glycodelin is important for helping with respect to the environment of the baby, of ensuring that it is in a good environment, and in particular, that it is in an immunosuppressive environment, so the immune system's not in there attacking the developing baby. We don't know, so this is a don't know as opposed to a no, we don't know which of these glycans are involved. However, nature has helped us a little to understand a little more of this system, because a significant number of women develop diabetes during pregnancy. This is called gestational diabetes. It's a bit like type 2 diabetes, but it goes away once the baby is born, so it is associated with pregnancy. When we characterized glycodelin A from these diabetic people, this is normal and this is the diabetic. What we noticed is that the diamonds, which are the sialic acids, are missing from the diabetic patients. So we know that sialic acid is important for immunosuppression because we also know that the diabetic GDA is no longer as immunosuppressive as the normal glycodelin. So now at least we have a little bit of insight into how this glycodelin is functioning as an immunosuppressive molecule. But we're no means well informed. However, we have another little bit of help, and that's from the guys in the audience. because you folks make glycodelin as well. So it's okay, you know, glycodelin's a wonderful molecule. You make it as well. And you make it in your reproductive tract, so it finishes up in the seminal fluid surrounding the sperm. Early on, before we had characterized the glycosylation, a lot of people were very worried about this because, as I said, glycodelin A, I didn't say it quite like that, but what in effect I said is it's a potent contraceptive. And the idea that a potentially potent contraceptive was bathing the sperm was not being sort of received very well by my male collaborators. But I said, don't worry, guys. I haven't done the sugar structures yet, but I'm absolutely sure they're going to be different. And as you can see from these pictures here, this is the girl, this is the boy. They are very different. What was particularly interesting in the male glycodelin was the presence of these sugars here, which have two of these triangles. These triangles are called fucos on the antenna. And that is called Lewis Y. It's another cluster of sugars with the name Lewis Y. Now, for many years, Lewis Y was thought to be a cancer antigen only. It had only ever been found on cancers. It is rarely ever found in women. And if it is found, it's mostly associated with cancers. In men, on the other hand, it is the most abundant cluster of sugars in your reproductive tract. So if we look at sperm, this is just simply human sperm, saying what kind of sugars are there. They're absolutely coated. And these sugars are known to be recognized by lectins of the immune system, for example, DC-SINE. We also know, as I've said, that Lewis Y is a major epitope in cancer. Now, you may not know what these pictures mean, but I think you can tell that that looks very different from that. What this is showing is the expression of Lewis Y on this extra tissue within the pancreatic cancer cells here. So, what does this all mean? I don't want to sum up saying that I've got a complete story, but what I want to sum up saying is that what in effect we're looking at here is the issue of self and non-self, and this dilemma that in the reproductive tract, there has to be a situation where the immune system is effectively being told, do not kill non-self. And in essence, this is associated, obviously, with reproduction, which is placentally associated reproduction, having the development of the embryo linked via placenta to the mother's bloodstream. And if you like, as this is a slide I've borrowed from Gary Clark, is an Achilles heel of the whole system. And the hypothesis here is essentially that the price we pay for having that long development of the baby is that we will have an immune system process of suppression which can be hijacked by parasites, for example, and also, in a sense, hijacked by aberrant cells in our own body, by cancer. So that is, if you like, the price we can pay. But on the other hand, I don't want to end on what might sound a bit depressing, because I think it does offer, if we understand it, quite exciting opportunities. So in principle, if we can fully understand these processes, we should be able to intervene and prevent, for example, the cancer developing, let's say, by switching back on again the ability of the immune system to kill the cells, but we really need to fully understand the processes. So in summary, what I've told you about today is that sugar recognition is incredibly important in cell-cell engagement. And what we know is that when cells come together, you can see the sugars meet the lectins and the whole cells light up. In other words, they send signals into the cell to tell those cells to behave in particular ways. Now, we know that's the overall, if you like, summary process, the headline process, but a lot of the detail is missing. And until we understand that detail, we won't be able to take this information as far forward as we would like. So in summary, I'd like to thank many of the people who've been involved in this work. These are people from the group currently and many people over the years. I want to flag up again the importance of Howard Morris, who has been my mentor all my academic life, two other senior members of staff in our group, Maria Panico and Stuart Haslam, and a whole range of students and postdocs here. And without all our collaborators worldwide, we wouldn't have been able to do any of what I've told you about today and any of the other things we've done. I do want you to note what this map is. It's taken off the web not that long ago, and it is the national happiness rankings of the world, and with the darkest blue being the happiest and the red the least happy. And you'll be pleased to know that Australia is completely dark blue. Sadly, although Ireland is dark blue, the rest of the United Kingdom is not quite making it to those happiness rankings. And thank you for your attention.
Thank you, Anne, for a very inspiring lecture. It certainly delivered a lot of messages to me personally about your career trajectory, actually. I'm pretty sure that young lady in that picture was you. Is that right?
Yes, it was.
I thought it might well have been. I think for me, as a scientist that has engaged mass spectrometry, the technique of accurately weighing molecules has been fascinating, and the development, as Anne has described, she's been there right from the beginning. So you can understand why her career trajectory has been as it is. She's developed so much and is recognised as being such an important player in mass spectrometry generally, but particularly one of the leading proponents of this technique in the world. It's great detective work. That's why I really look at it in terms of how you can understand these big biopolymers of sugars and drill down to the nth degree about saying, well, how are they connected together? More importantly, linking that then to disease. I think it's absolutely fascinating. So congratulations, Anne, for that. I have a question, a scientific question, but I want to ask the audience first. We'll just have a moment for questions, if that's okay, before we retire to celebrate this wonderful lecture. So I'll open it up, first of all, for questions from the audience. Catherine.
Yeah, thanks for that. I really enjoyed the mass spec part
Thank you.
of that. So I suppose following on from Mark a little bit, do you take those mass spectra and sit down and investigate manually, or is there software these days?
So perhaps before you answer the question, I might just repeat it so we capture the question. Mm-hmm. The question simply was, is that, well, when you collect all of this wonderful data, all those wonderful spectra that you saw during the talk, do you have software that can automatically analyze that, or do you have to go through this bit by bit yourself?
It's a mixture, actually, is the answer to that. So for 25 years, we looked at it manually. In fact, in the very early days, where for the first sort of 8 or so years of my academic career, the way the data was recorded was on photographic chart paper. It was exactly the same kind of photographic paper that you could buy from a photography shop. Rolls of Kodak, actually, we used to get paper. And we just simply rolled it out, and we counted from 28, which is the mass of nitrogen, there's always a bit of nitrogen gas that ionizes. And we counted it 1 mass unit at a time up the spectrum. Then computers came along that were actually able to do the counting, if you like. And then the interpretation for certainly 10 or 15, 20 years was entirely manual, until we did get a bit sort of bored with the fact that we were getting by then too much data to do that really realistically. So we there is software, but we had a very talented bioinformatics postdoc who wrote some really nice software which is open access on the web. And what that software does effectively is to create those cartoons using the information. It is not completely automated, and that is deliberate because we don't want people to be misinterpreting their results. There's a variety of other software that's available on the web. So it is in between being manual and automated. But I would also say that the most exciting bit is getting that data and then being able to work out these ions. The zona pellucida egg data was just absolutely amazing. There was no way we used software to interpret that. We did that manually.
Any other general questions that people would like to ask? Then I'll turn to my final question. If not, I'll ask one final question then. So we know that men are becoming irrelevant. The Y chromosome is shrinking, we know that. It's going to be completely gone eventually. I have no doubt about that. Well, according to my colleagues in the Academy of Science here, that's what they tell me. The question I have is going back to your sperm glycan carbohydrate story and the egg engagement. We know that actually there's an enzyme in the sperm, in fact in the cap, in the top of the sperm, that apparently is secreted, that actually chops sialic acids away, that wonderful sugar that you spoke about. How does that relate in terms of the story about engagement of the sperm?
Okay, so what I will not perhaps have shown you as well as I should have done to answer this question is that the egg is a very, very tiny thing. It's about the size of a full stop if you think about an ordinary size pen or pencil as opposed to making any size on a computer. But surrounding it is this sugar coat, the xenopellucida. So when the sperm arrives at the egg, it engages initially with this sugar coat. After that, there is a reaction which is called the acrosome reaction, which is what Mark is referring to. And what that essentially means is that inside the sperm is a pretty boring cell, actually. It really has, or those nice cells I showed you in cartoon, which had all of these different compartments inside to show they were interesting cells. The sperm doesn't have all of that. It just has a little package of DNA, which is sort of important. But then it has a bag of enzymes, which is called the acrosome. And yes, the first thing that happens upon that engagement, again, not fully understood how it happens, but is for the, if you like, the front of the sperm to blow apart and for these enzymes to spew out. And their role is to do all sorts of things, including making a way for it through that sugar layer. And although we don't understand whether or not one could argue that the sialidase is needed to make sure other sperm don't engage, because that's the critical thing. Remember that these millions of sperm start off, they finish up 2 days later, maybe hundreds, but there's still hundreds, only one is making it. So that it is very important that you don't get the egg being fertilized by more than one sperm, because that can create problems. So that is almost certainly the reason for those enzymes spewing out, a combination of it then eating its way into the egg, but also to make sure that other sperm are not going to fertilize. Sialic acid is really, really important.
Yes, absolutely right. I completely agree with you. So before I invite Susan Gallagher up to award you this wonderful plaque in recognition of your outstanding lecture, I'd like all of us to join in thanking Anne for what I thought was a very articulate and excellent lecture.